Assalamu alaikum. My dear students, we are still in chapter 3 adaptive immunity, part 3 humoral immunity. Now I'm going to remind you with some information, basic information that I explained before in the previous slides of this part. So basically we have two types of immunity. These are the innate immunity and the adaptive immunity or acquired immunity. The innate immunity is non-specific and has its own mechanisms. We discussed this before in details. The adaptive immunity is composed of two wings. These are the humoral immunity and the cell-mediated immunity. Let's focus on humoral immunity. Humoral immunity of the human body is mediated by the B lymphocytes. The B lymphocyte originate from the bone marrow, as all other cells of blood. Inside the bone marrow, the B lymphocyte go through partial maturation. So the B cell go through multiple stages inside the bone marrow as part of the partial maturation. One of the important events that happens to the B lymphocyte inside the bone marrow is the formation of the B cell receptor. The B cell receptor is a membrane-bound immunoglobulin composed of 4 protein chains. These are 2 heavy chains and 2 light chains. Then the B lymphocyte go through 2 major main checkpoints inside the bone marrow. These are positive selection and negative selection. So the positive selection of the B lymphocyte inside the bone marrow is to check whether the receptor, the B cell receptor, is working properly or not. And the negative selection that happens to the B lymphocyte is part of the B cell central tolerance to check whether the B lymphocyte is self-reactive or not. Then the B lymphocyte reach immature stage containing IgM receptor. Migrate from the bone marrow to the secondary lymphoid organ as immature B lymphocyte. Then the immature B lymphocyte complete the maturation process inside the secondary lymphoid organ. Once the B lymphocyte reach the secondary lymphoid organ, for example the spleen, enter the spleen, they are said to be T1 B cell. Transitional 1B lymphocyte, and then the transitional 1B lymphocyte transform into transitional 2B lymphocyte. Furthermore, the transitional 2B lymphocyte differentiate into one of 2 types: marginal B lymphocyte or follicular B lymphocyte. These marginal and follicular B lymphocytes are naive, mature B cell. They are now ready to recognize foreign antigens. They circulate inside the secondary lymphoid organ, doing the job of immune surveillance, looking for appropriate antigen to recognize. Once they find the antigen, they can recognize it in unprocessed free form. So the B cell can recognize unprocessed antigen, free form antigen. The B lymphocyte doesn't need any kind of antigen presentation. Once they recognize appropriate antigen, the B cell go through the series of activation. So activation can happen in 2 different ways, as we said before. The first is dependent upon helper T lymphocyte. The other is independent on T cells. The first is said to be T cell dependent B cell activation. And in this case, the B cell depends on cytokines and signals from helper T lymphocyte. or CD4 positive T lymphocyte. While the other type is T cell independent activation, and it happens because of the presence of T cell independent antigens. And we said before that, for example, lipopolysaccharide of gram-negative bacteria, which leads to the activation of B cells independently of T lymphocytes. Okay, once the B cell is activated, they go through a process of differentiation. Of course, activation of B lymphocyte, recognition of the antigen and the activation of B lymphocyte is based on clonal selection. Then the selected clone will go through a process of clonal expansion, differentiation, to increase the number of the B lymphocyte clone. Then they go through differentiation. The differentiation will yield or produce two cell lineages. These are plasma cells and memory cells. The plasma cells are the effector cells of the humoral immunity. They are the cells that can produce and secrete the secreted form or soluble form antibodies that will react against the antigen, helping in elimination of the pathogen from the body. While the memory cells are dormant, some of the memory cells may stay in the secondary lymphoid organs, others may migrate to the bone marrow. They will stay dormant in the body for years or even the lifetime. They will never be active again unless the same exact antigen reinfects the body again. So in this case, the responding cell will be the memory cell. In case of reinfection with the same antigen, I mean in the secondary infection. The secondary responder cells will be the memory cells. They will do vigorous rapid response against the antigen, preventing the pathogen from inducing any disease in the body. The antibodies or the immunoglobulins. So the antibodies or immunoglobulins are glycoproteins belonging to the superfamily Igs or immunoglobulin family. They are a type of gamma globulins. You know that globulins are alpha, beta, contains alpha, beta, and the gamma, and these antibodies or immunoglobulins belong to the gamma globulin fraction. Each antibody monomer, the basic unit of antibody, is Y-shaped structure, contains 4 protein chains, 2 heavy chains and 2 light chains. You can see this here. I'm going to point. We have 2 heavy chains. See the long one, 1 and 2, and 2 light chains, this one and this one. So at the tip of the antibody, there are 2 sites here. We call them as the paratop. The paratop is an antigen binding site. This red one is the antigen. The antigen contains epitope, and the paratop of the antibody can bind to the epitope of the antigen. So the paratop is part of the antibody, while the epitope is part of the antigen. Like lock and key and lock. Each pathogen contains an antigen or more, may contain one or more antigenic determinants or epitopes on the surface of the antigen, part of the antigen, of course. So each epitope has its own specific antibody for binding. So if one virus or bacteria contains an antigen which has more than one epitope, it means that they will trigger the formation of more than one type of antibodies, depending on the number of epitopes on that antigen. Valence of an antibody means the number of antigen binding sites of this antibody. Most antibodies are bivalent because most of them are monomers. A monomer antibody is Y-shaped, containing two antigen binding sites, bivalent. But for example, the IgM contains 5 monomers, pentamer. Each monomer contains two antigen binding sites, so the total number of antigen binding sites of the IgM will be 10, decavalent. The ability of the antibody to communicate with other components of the immune system is mediated by the Fc part or the Fc region of the antibody, the stem of the antibody. So the tip of the Y shape is for binding the antigen, the stem is for communication and the mechanisms of the antibody. As we know that the antibodies are of two forms, physical forms. The first is the soluble form, the second is the membrane-bound form. The membrane-bound form antibody forms the B-cell receptors by which the B-cell can recognize antigen. While the soluble form is produced by the effector cells, which is the plasma cells, to react with the antigen and eliminate and help eliminating the pathogen. So once the B-cell receptor can find the appropriate antigen and recognize this antigen, the B-cell will go through activation process, clonal selection, clonal expansion, differentiation, producing plasma cells and memory cells to fight against the antigen. Okay, now I'm going to explain the antibody structure. So the antibody is Y-shaped. The monomer antibody is Y-shaped, as you see in this graph. This Y-shaped monomer is composed of two heavy chains. You see this blue, one heavy chain, and this one is another heavy chain, and two light chains. So the antibody monomer is composed of two heavy chains and two light chains. So there are 4 polypeptide chains constituting the monomer antibody, 2 light chains and 2 heavy chains. The light chains are the smaller chains. These are the smaller chains. They are composed of about 25,000 Daltons. While the H chains are the large chains containing a higher molecular weight, about 50,000 Daltons. The antibody contains variable region and constant region. So the variable region is this one, contains one arm of the light chain and one arm of the heavy chain. Half of the light chain and 1/4 of the heavy chain. This is the variable region. See, inside this circle is the variable region. So the variable region is the end of the Y arms, and it contains the antigen binding site. So the antigen binding site is the end of the variable region, by which the antibody can bind the epitopes of the antigen. See this part? This is antigen, and these are the epitopes, and they are binding to the antigen binding site of the antibody. These two antigen binding sites are identical on the same antibody, but they are different from one type of antibody to another type of antibody. The other region is the constant region. The constant region is the base of the antibody, the stem of the antibody. So it contains about half of the light chain and all other parts of the heavy chain. For the heavy chain, we assume that it contains at least 3 segments, each chain, 3 segments of the constant region. So we call it C heavy 1, constant heavy 1, CH2, CH3. So this part is the constant region of the antibody. The constant region, which is the stem of the antibody, is very important because it can bind to complement or cells. And by the constant region, the antibody can do the mechanisms to do the reactions against the antigen. So we have what we call FAB, fragment antigen binding region, FAB region, or fragment antigen binding region. This part is the FAB, or the fragment antigen binding. As you see, the fragment antigen binding region is composed of one variable and one constant part of the light chain plus one variable and one constant part of the heavy chain. So it is composed of one constant and one variable domains of each heavy and the light chains of the antibody. While the end is called the paratop, the end terminal or the amino terminal end of the antibody is called the paratop. So the paratop is shaped at the amino terminal end of the antibody monomer by variable domain from the heavy and the light chain. The paratop is the part by which the antibody binds to the epitope of the antigen. And the antibody also contains the constant region. The constant region. All this is the constant region. The constant region determines the mechanisms by which the antibody can do the reactions against the antigen or destroy the antigen. And we said before that by the constant region, the antibody can communicate with other components of the immune system. Actually, antibodies are divided into 5 major classes depending on the types of heavy chains. So according to the type of heavy chains, there are 5 classes of antibodies. These are gamma, delta, alpha, mu, and epsilon. And in English, we say immunoglobulin IgG, IgD, IgA, IgM, and IgE. So we have 5 classes of antibodies, 5 classes of antibodies according to the type of heavy chain. These are G, D, A, M, and E. Immunoglobulin G, immunoglobulin D, immunoglobulin A, immunoglobulin M, and immunoglobulin E. Okay, to remind you, I'm going to read the notes to remember what we have said before. So number 1, though the general structure of all antibodies is very similar, a small region at the tip of the protein is extremely variable. The variable region, the antigen binding region or the antigen binding site, allowing millions of antibodies with slightly different antigen binding site to exist. This region is known as the hypervariable region. So that the tip of the antibody is hypervariable. They are very different among different antibody types. Each of these variants can bind a different antigen. يعني ربنا سبحانه وتعالى مكن خلايا بي من إنتاج different antibody types، وبالتالي different receptors، على اعتبار إنه الانتيبوديز عبارة عن نوعين. النوع الأول عبارة عن بي سيل ريسبتور وده ممبرين باوند فورم، والنوع الثاني سكريتد فورم. علشان تستطيع الخلايا المناعية بي أن تقوم بعمل recognition ل different wide variety أو different types of antigens، وبالتالي تبقى قادرة على التعامل مع different types of pathogens. كمان الخلايا المناعية بعد ما يحصل لها الخلايا المناعية بي بعد ما يحصل لها activation وتتحول إلى بلازما سيلز، هتكون وتفرز different types of secreted form antibodies. It depends على إيه؟ It depends على التايب of B cell clone. تمام؟ وطبعا معتمدة على نوعية الانتيجين، على اعتبار إن في ماتشنج ما بين الانتيبودي اللي هيبقى يا إما بي سيل ريسبتور أو سكريتد فورم مع الابيتوبز بتاعت الانتيجين عشان يقدر يعمل بايندنج، وبالتالي يقدر يعمل التفاعلات بتاعته معاها، وديت رحمة من الله سبحانه وتعالى. هقول تاني. So the region is known, this region اللي هي الانتيجين بايندنج سايت بنسميها الهايبر فاريبل region. Each of these variants can bind to different antigen. This enormous diversity of antibody paratopes on the antigen binding fragments allow the immune system to recognize wide variety of antigens، وبالتالي wide variety of pathogens، وبالتالي يبقى الجسم محمي بنوعيات مختلفة من الخلايا قادرة على التعامل مع أنواع كثيرة من الميكروبات. The large and diverse population of antibody paratope is generated by random recombination events of a set of gene segments that encode different antigen binding sites or paratopes. أفكركم لما اتكلمنا عن الدايفرستي in the immune system لو فاكرين، وقلنا إنه الدايفرستي بيحصل نتيجة تكون different receptors، وإن الديفرنت ريسبتورز دي بتتكون نتيجة ريكومباينيشن، راندوم ريكومباينيشن ما بين 3 جين سيجمنتس في دي جي. اتكلمنا في الحكاية دي أكتر من مرة وعرفناها. ولما جينا نتكلم عن الديفلوبمنت اوف ذا بي lymphocyte inside the bone marrow، فاكرين لحد ما تصل إلى immature cell، اتكلمنا عن ستيجز وقلنا إنه أثناء هذه الستيجز بيتم recombination of those gene segments ال V وال D وال G على بتاعت الهيفي تشين، وال V وال J على اللايت تشين، وبالتالي يتكون الانتي بودي اللي بيستخدم كريسبتور. Okay. Now we will talk about the structure and the characteristics of antibody isotypes. These are the 5 isotypes that we mentioned before: IgM, IgD, IgG, IgA, and IgE, according to the heavy chain differences. Each of those isotypes contains its own characteristics and its own functions or rules. Number 1, the IgG, the most important and the dominant immunoglobulin in our bodies. The IgG molecular weight is about 150 kilodaltons. It is a monomer. So the IgG is a monomer composed of just 1 unit of Y shape. It contains 4 subclasses: IgG 1, 2, 3, and 4. The percentage in serum is about from 70 to 80%. It is dominant, really dominant antibody. It is located in blood, lymph, intestine, in all other body fluids, and also in some tissues. You can find IgG antibodies. The half-life in serum is about 23 days. The functions of the IgG is, it has a variety of functions. It's important, really important. For example, it enhances the phagocytosis, because the IgG antibody can do opsonization. It is a very important opsonizing agent. It is working as opsonine. Neutralize toxins and viruses. You know that viruses can infect the cells of the body through attachment with certain receptors of those cells. The IgG antibody prevent this attachment by blocking the receptors of the specific for those viruses, and also can neutralize toxins by doing the same. They may block the receptors of those toxins, or they may change the configuration of the toxins. So in any way they can neutralize toxins and viruses, preventing viruses from infecting cells, preventing toxins from toxicity of those cells. It is the only immunoglobulin that is transferred across the placenta from mother to fetus. So it protects the infant until its own immune system is functional. And it circulates in blood and can easily exit to the tissues. This is a brief summary about the IgG antibody. Number 2, the IgM antibodies. So the IgM molecular weight is about 900 kilodaltons. It is pentamer, composed of 5 monomers, 1, 2, 3, 4, 5, as you see in this graph. The percentage of serum is about 5 to 10%. The location is in blood, lymph, B cell surface as a receptor, and in this case it is a monomer. The half-life is about 5 days in serum. The presence of IgM in newborn indicates intrauterine infection. Effective against microorganisms and the clumping antigens. So the IgM, because it is pentamer, pentamer means it contains 10 antigen binding sites, decavalent. So it is important, very important in reactions to clump the antigens, agglutination reaction, for example. It is the first antibody to be produced in response to mild infection. So the first infection will yield IgM antibodies in humans. It is found in circulatory system with IgG always. IgG and IgM can activate the classical complement pathway. If you remember, we said that 2 molecules of IgG or 1 molecule of IgM react with the antigens of the pathogen, for example bacteria, can activate the classical pathway, producing finally the membrane attack complex, or the MAC, that cause lysis of the pathogen or the microorganism, the microbe. The IgM has lower affinity for antigen than IgG. IgG is has more affinity, more active against antigens, but the IgM is more important in case of agglutination reaction or clumping of antigens because of the decavalency of the antibody IgM. Number 3, the IgA. The IgA molecular weight is about 320 kilodaltons. It is dimer, composed of 2 monomers. The percentage of IgA in serum is about 10 to 15%. The IgA occurs in 2 forms. These are IgA1 and IgA2. The half-life in serum is about 6 days. It provides the localized protection on mucous surfaces of the body. Can be found in mucus, tears, saliva, sweat, blood, human milk, and intestinal fluids. The IgA protects surface tissues and prevent adherence of microorganisms. This is why we said that it provides a localized protection on mucous surfaces of the body. It is also important in respiratory, gastrointestinal, genitourinary tract infections. The IgA in breast milk protects the gastrointestinal tract of the neonates. Number 4, the IgD. The IgD molecular weight is about 180 kilodaltons. It is monomer. Percentage in serum is very low. It is less than 1%. Location of IgD is on B cell surface. It works as a receptor of mature B lymphocyte, as we said before, naive B cells, and very low amounts in blood and lymph. Half-life in serum is 3 days, very susceptible to proteolytic attack. It is susceptible to lysis. The IgD non-function is to work as a B cell receptor to initiate an immune response. Number 5, the IgE molecular weight is about 200 kilodalton. It is monomer. Percentage in serum is about 0.002%. It is very, very small amount. The IgE antibody can bind by the Fc portion to the mast cell and the basophil in the body, and it is responsible for allergic reactions and the initiation of inflammatory reactions in the body because of its ability to bind to the mast cells and the basophils, allowing those cells to secrete more mediators like all the mediators, histamines, leukotrienes, heparins, chemotactic factors, cytokines, and all those types of mediators. Maybe sometimes it is involved in lysis of parasitic worms. Now we are going to talk about immunoglobulin class switching or isotype switching. Actually, the isotype switching is to change the ability of the B lymphocyte to produce different isotypes. For example, producing IgG instead of IgM. To do so, the B lymphocyte change the constant region of the antibody heavy chain, but not the variable region of the heavy chain. The variable region of the heavy chain stays the same with no change. By this way, the antibody retains the same specificity, no change in the specificity of the antibody. Okay, let me explain this very simple drawing. This simple drawing shows us the activation and the class switching of B cells. At first, the antigen-presenting cells present antigen to the T helper cells. So this is an antigen-presenting cell using MHC class II molecule with peptide antigen, present the antigen to the helper T lymphocyte. So then the T helper lymphocyte activate. So number 2, B7 is expressed and interacts with CD28 activating T helper cell. These are co-receptors for activation of T helper cells. Then the T helper cell is activated. So activated T helper cell interacts with B cells via the CD40 ligand, activating B cells to proliferate, differentiate, and secrete antibodies. As you know that B cell and T gets help from T cells through the CD40 and the CD40 ligand. CD40 on the B cell, CD40 ligand on the T cell. Also, the activated T cell will secrete different cytokines. So the secreted cytokines will determine the class switching mechanism. So, for example, the IL-5 will switch from IgM to IgA, while the IL-4 will switch from IgM to IgG or IgE. We will explain this in a table in the next slide. At first, the pathogenic infections activate the B lymphocyte to produce IgM. We said before that IgM is the first antibody isotype to be produced in mild infections. Then the B lymphocyte gets help from the helper T lymphocyte, the CD4 positive T lymphocyte. This help is in the form of signals via the CD40 molecule, which is one of the important co-receptors for the B lymphocyte, or through cytokine receptors. So that CD4 positive T lymphocyte produce different interleukins that may affect the ability of the B lymphocyte to switch the isotype from IgM to IgG, IgA, or even IgE antibodies. The change will be in the Fc region of the antibody only. There is no change will happen in the variable region. Changing the Fc region means changing the mechanisms by which the antibody will react against the antigen. But changing the variable region means changing the specificity, which is very dangerous, will never happen. So the specificity of the antibody will be the same, so that the antibodies produced by the B lymphocytes, even switched antibodies produced by the B lymphocyte, will attack the same antigens specifically, but with different mechanisms. This is isotype switching. To understand the class switching, look at this drawing. At first, the B cell will produce IgM antibodies in case of mild infection. So when the B lymphocyte encounter a pathogen for the first time, they produce IgM. Then the B lymphocyte will get help by the CD4 positive T lymphocyte, the T helper cell. This help is in the form of different cytokines, allowing the B lymphocyte to produce different isotypes. For example, the interferon gamma produced by the Th1 will affect the B lymphocyte to switch from IgM to IgG1 and IgG3. It means that interferon gamma will trigger the formation of IgG subclasses instead of IgM. So those IgG subclasses will do so many functions. For example, opsonization, enhance the phagocytosis, complement fixation or activation, and neonatal immunity, because the IgG subclasses are the only isotypes of the antibodies that can cross the placenta. Other interleukin 4, IL-4, which can switch the IgM formation to the IgE and IgG4. IgE antibody is important in immunity against parasitic worms, for example, helminthes. Also, the IgE Fc portion can bind the receptors of certain receptors of the mast cells, leading to degranulation and releasing mediators, leading to immediate hypersensitivity. Also, if the B lymphocyte gets help by other cytokines like the tumor growth factor beta, the April or the BAF or any others, may change the IgM to IgA, which produce mucosal immunity. See also this graph. In the primary response, at first IgM is produced and then IgG antibody. But in secondary immune response, the IgG antibody is produced by huge amounts. And we can summarize the effect of different cytokines on B lymphocyte to switch the isotype production. See, Th2 helper lymphocyte, I mean Th2 cells, can produce IL-4 to affect the B lymphocyte producing IG1, IG4 and IgE. Or the Th2 helper cell can produce IL-5 to affect the B cells switching from IgM to IgA. The interferon gamma will trigger the formation of IG3 and sometimes IG1. The TGF beta, tumor growth factor beta, will trigger the formation of IgA. The IL-10 from T regulatory cells will trigger the formation of Ig1 and Ig3. Now we reach the biological activities of the immunoglobulin molecules, I mean the functions of the antibodies. What are the mechanisms that the antibody can do to help elimination of different pathogens by the action with their antigens? Number one, agglutination. Agglutination means that the antibody can cause the antigen or the microbe, because the antigen is part of the microbes, to clump together, so prevent the dissemination of the microbe and enhance the removal of the microbe by phagocytes. The IgM is the best, more effective antibody to do this job, because the IgM is decavalent, contains 10 antigen binding sites, so it can do clumping of the antigen better than IgG, which is only bivalent. Hemagglutination means the agglutination of red blood cells, for example the ABO blood types, the A and B antigens, self-antigens, to detect influenza and measles viruses, and this is very important. Number two, opsonization. Antigens, which is part of the microbes, is covered with antibodies. So the antibodies here mark the antigens that enhance the ingestion and the lysis by phagocytic cells. See, this is the microbe. The microbe is binded by the antibodies. So the antibodies are binding to the antigens of the microbe. And in the same time, the antibodies are attached to the phagocytic cell by their Fc portion, allowing the phagocytic cell to phagocytize this antigen, this microbe, easily. So binding of antibody-coated pathogen to Fc receptor on phagocyte is to facilitate phagocytosis. It allows the phagocytic cell to engulf the pathogen easily. Number three, neutralization. So neutralization is best done by IgG and sometimes IgA antibodies. So the antibodies can inactivate viruses from binding to their surface receptors, and sometimes the IgG antibodies can neutralize toxins by blocking their active sites. So the IgG antibodies or sometimes the IgA antibodies prevent attachment to cell receptors, because the viruses need to attach to cell receptors for infection, and the toxins need to attach to cell receptors to toxify the cells, to make toxicity of those cells. The IgG antibody rule, the IgG or IgA antibody rule in this case, is to prevent this attachment of viruses or toxins. So they can prevent infection by viruses, they can neutralize toxins, sometimes, for example, tetanus toxins. Number four, the antibody-dependent cell-mediated cytotoxicity. This happens to destroy large organisms, for example, helminthes or worms, parasitic worms, because these are large organisms. So the antibodies coat these organisms, allowing other immune cells, non-specific cells, cells of the innate immunity, to attack those organisms, worms, and destroy them. Number five, the complement activation, and we discussed this in details before, and you know that IgG, 2 molecules of IgG or 1 molecule of IgM will trigger the classical complement activation, leading to lysis of the pathogen, or leading to enhancing inflammation to eliminate the pathogen. Number six, immobilization and the prevention of adherence. For example, immobilization of bacteria. It means preventing the bacteria from free movement. And this happens when certain antibodies can attach the flagella or pili. By this mean, the antibodies interfere with the mobility and the attachment of bacteria, allowing phagocytic cells to engulf them more easily. Number 7, precipitation. Precipitation means precipitation of soluble antigens, and this will allow the phagocytosis to get rid of them or remove them more easily. So precipitation is best done by IgG antibodies. Virus blocking, and we said that before. Number 9, mucosal protection, and this is provided mainly by the IgA antibodies. And number 10, conferring immunity to the fetus by crossing the placenta, and this can be done by the IgG antibodies. Number 11, newborn protection by passage of IgA antibodies in colostrum and milk. Antibodies. So monoclonal antibodies are very specific antibodies, can be produced by just one clone of B lymphocytes, artificially produced in large amounts, and be used in diagnosis and therapy. In diagnostic uses, they are used for detection of CD markers on lymphocytes, different HLA antigens, viral antigens, tumor antigens, and the measurement of hormone levels. In therapeutic uses, they are used as anti-tumor therapy, immunosuppression to prevent graft rejection, treatment of drug toxicity, passive immunotherapy, for example, against viral infections or diseases, and the prevention of Rh incompatibility.